Bismillah Rahman Rahim. Welcome again, guys. Welcome to another video lecture by Biology. Today in this video lecture, we are going to talk about termination stage in prokaryotic cell. Termination of replication inside the prokaryotic cell or inside the E. coli cell. In the previous video lecture, if you guys remember, we talked about the DNA replication, basics of DNA replication. We talked about the proteins which are involved in DNA replication in prokaryotic cell. And we also talked the initiation, elongation in prokaryotes as well. And we talked different kinds of enzymes that are involved in each stages. Okay, this is the last video lecture of the DNA replication in case of the prokaryotic cell. And in this video lecture, we are going to talk about termination. But before going to explain the termination, you guys need to understand about the 3 video lectures of DNA replication which I already explained. The first video is the protein which are involved in DNA replication. The second video you need to understand the initiation, and the third video you need to understand elongation. And then you will come to this video lecture, then you will get the whole concept about the DNA replication in case of the prokaryotes. Okay, so let me clean the whiteboard and let me explain the DNA replication. Let me explain the termination stage in DNA replication. So let me write termination. Termination of DNA replication in prokaryotes, usually in E. coli cell. Now, what is termination? Termination is that stage in DNA replication that when the nucleotide are added, that during elongation stage, when the nucleotide are elongated, are added, and when the strand are elongated, then the replication fork and helicase and the 2 bubbles meet at a specific point, at a specific complex which we call that TER and TUS sequence and TUS protein complex, due to which the replication are stopped and the new DNA strain are then separated from each other, from the old strain. This is called as termination, and the replication are stopped at that specific point. We call that termination stage in DNA replication in case of the prokaryotes. It is also involved in eukaryotic cell, but inshallah we will explain DNA replication in case of the eukaryotes as well. Now, what kind of proteins we need in termination? In termination, we need a protein we call that TUS protein. We also call that TUS stand for termination utilization site or termination utilization substance. Okay, and the TUS protein can be bind with the TER sequences. So we have another sequences which can stop the replication. We call that TER sequences. TER sequences, which is almost 24 base pair long. Okay, TER sequences is usually present, means opposite to the origin of replication. We will see it later. Okay, so at TER sequences and TUS protein, they make a complex which we call it TER sequence TUS protein complex, and at that complex the replication are stopped and then the DNA are separated from each other. Okay, then we have another enzymes which we call that topoisomerase 4. When the replication process are completed, then topoisomerase 4 can cleave the 2 strands from each other and separate it. This is called segregation. They can segregate the 2 strands. Okay, so topoisomerase. Now, how exactly the termination are happen? Now, to explain the termination, let me draw again a simple diagram of the bacterial chromosomes, and let me explain the termination. So here I will draw the 2 replication bubble and we will draw the exact structure. So let's say this is the one replication bubble and this is the another replication bubble. Okay, so now we have two replication bubble. One replication bubble, replication fork and helicase. All these things can move in this direction, while the other replication bubble, fork and helicase or DNA B protein can move in this direction. So we can say at that specific position, that is called as origin of replication, which we call it ORIC. So actually the DNA can replicate in bidirectional form. Bidirectional. Okay. Now both fork can meet at a point which we call that ter and tus complex point. Let's suppose at that specific area. So we call that ter tus complex. And at that specific position the DNA replication will be completed and then the DNA will be separated. Okay, then what actually happen? We have two let me once again tell you. We have two replication bubble. This is what replication bubbles, and this is the replication fork, and as well as we have the helicase protein, or we call it DNA B protein. So two replication bubbles are moving in opposite direction and they meet at a specific point which we call it ter and tus complex. Ter is actually 24 base pair sequences and we have almost 5 ter site. Okay, we have almost 5 ter site at opposite direction. So let me draw here the first, then we have second, third, fourth and fifth. Similarly we have also at that specific position 1, 2, 3, 4 and 5. Okay, so these are the ter sites having 24 base pair sequences. So each ter site have 24 base pair sequences, and moreover each ter site contain the ter sequences or the ter genome, we also call it ter genome. These sequences cannot terminate the replication by itself. They cannot terminate the replication process. They need a protein which we call a tus protein by the complex, then they can terminate the replication. Then they can separate the two strand. Not separate the two strand, we can say then the replication process will be terminated or replication process will be completed or end up. Now we call that protein, at that specific area we have what? We have the proteins which we call that. So let's say these are the protein. These ter sequences have two end. So let me draw a one ter sequences here. So this is the one ter side. So they have two sides. One is they have two end. Okay. So this ter side have two end. One we call that a permissive end. Permissive end. They can allow the DNA or they can allow the replication fork to passes, and the other is called as non-permittive end. They can stop the replication bubble. Okay. Permissive end can carry on the replication and they permit the replication bubble to passes, while the non-permissive end can prevent the replication bubble. They cannot allow the replication bubble to passes. Okay. Now what actually happen? Whenever the replication bubbles are moving and whenever they reach to the permissive end, remember, whenever they reach to the permissive end, then they can remove out this tus protein. So the TUS protein are then removed. These are the permissive end. Now at right side, the permissive end is present at the left. At left side, the permissive end will be present at the right. Okay. Now whenever the replication bubbles are moving with the permissive side, so then they can dissociate and remove the TUS protein. So whenever the TUS protein are removed out, so then the replication bubble is moving and replication is carry on and the elongation is carry on. So whenever they reach to this complex, TER and TUS complex, then at that specific area, actually the TUS is acting as a clip. Okay, they are just like in a ring form, and they can cannot allow the DNA or they cannot allow the helicase to move in the opposite direction. Let's suppose the helicase can move in this one direction. This is, let's suppose, the helicase that are move in this direction, but they prevent the helicase to move in the backward direction. Okay, this TUS protein can also flip two base pair nucleotide, and whenever they flip the two base pair nucleotide, then the replication forks are not going on, or we can say the helicase protein or DNA B protein cannot be passes. So let's say again this is the DNA strand. Okay, and here we have the DNA B protein or helicase is moving, and these are the hydrogen or nitrogenous bases, or these are the hydrogen bond. Okay, then at that specific position, two or one nucleotides are flipped. Okay, so these are the TUS protein. They can flip the two base pair or one base pair, usually cytosine and guanine can flip up. Whenever each nucleotide are nitrogenous bases or nucleotide are flipped, then the helicase cannot be moved. Whenever the helicase are not passes, so then the termination process, so this is the termination process, so then the replication process are stopped. Okay, and then the strands are separated. So this is actually the theta mode of replication. Theta mode of replication. No, why? Because they can make a shape like that. As you are watching, they can make a shape just like that. That's why we call that the bacterial chromosomes can be replicated through the theta mode of replication. But this is the bacterial chromosome. This is not the plasmid. Plasmid can be replicated by rolling circular replication method, like that method. Okay. These 5 sites which I write here, these 5 sites are named as F, G, F, B, C. Okay, and these 5 sets are called as, the name of these 5 sets are H, I, A, D, E. So these are the 5 sites. Okay, now let's suppose the replication bubbles are moved at that specific position, and they are passes, and the proteins are removed out, and then they reach to a complex. Then the strand are jumped like that, so then we get a strand like in this position, which is used as a template, and a new daughter strand is also synthesized. This is the new strand. Okay, so then what actually happen, this new strands are then jump like that, so we get the structure like in this form. So this is the new strand which is formed, and this is the parental strand. So then what actually happen, we get 2 strand. So again this is the daughter strand which is synthesized, and this is the parental strand. So this process is also called as semi-conservative. This is actually a semi-conservative DNA replication. Why this is called semi-conservative? Because in this case, the one strand is conserved. Okay, the one strand is conserved by the other new, and the other new strand is formed. So that's in a sense about the termination inside the prokaryotes. Okay, that's all about the DNA replication inside the prokaryotes. I hope you get idea about that, how the DNA replication are happen inside the prokaryotes. So if you have still any kind of question in your mind, you can write it into a comment. Inshallah, I will try my best to give you answer as soon as possible. If you like this video, so make sure to hit the like button, share this video, and also subscribe the channel. Inshallah, in some other video lecture, we are going to talk about the DNA replication in eukaryotic cell. Okay, so thank you so much for watching.